Hello everyone, myself Mr. Jangle Sanket Surendra, lecturer from Mula Education Society's Mula Rural Institute of Pharmacy, Sone. Today I am going to discuss the chapter Sterile Products. This chapter is a very interesting and important chapter in a pharmaceutics too. This chapter is generally discussed for the second year diploma in pharmacy students. Now we will start with the sterile products. Contents This chapter contains the introduction of parenterals or sterile dosage forms, advantages and disadvantages of parenteral preparations, route of administration of parenteral products, general requirements for parenteral dosage forms, types of parenteral preparations, formulation of parenteral preparations, processing of parenteral preparations, and evaluation of parenteral preparations. Learning outcomes. At the end of this lecture, the learner will be able to understand the introduction of these sterile dosage forms, advantages and disadvantages of parenteral preparations, routes of administration of parenteral products, general requirements of parenteral dosage forms, types of parenteral preparations, formulation of parenteral preparations, processing of parenteral preparations, and evaluation of parenteral preparations. Now we will see the introduction of sterile dosage forms or parenteral preparations. The parenteral preparations are those pharmaceutical products that are given by other than oral routes. Transfusion fluids and injections are the parenteral preparations. Injections are the sterile solutions or suspensions of drugs in aqueous or oily vehicles means used for introduction into the body by means of an injectable needle under or through one or more layers of the skin or mucous membrane. This injection should be sterile, isotonic, and free from foreign particles such as dust, fibers, etc. They should be free from foreign particles too. Then, for example, an oily suspension meant for intramuscular injection may be very dangerous if it is administered by intravenous injection. Similarly, those potent drugs which are required to be given through intramuscular injection may prove very fatal or serious if it is given by intravenous route. Now, the advantages of parenteral preparations. The first advantage is the drugs which cannot be taken or administered by oral route can be administered by this route. Second one, the onset of action is very quick. The patients who are vomiting or unconscious cannot take the drugs by oral route. In such cases, the drugs can be administered by this route. The drug action is quick and rapid. Drug action can be prolonged by modifying the formulation, means it can be increased for a longer time. Transfusion fluids containing nutritives like glucose and electrolytes such as sodium chloride can be given by this route. These are the advantages of parenteral preparations. Now we will see the disadvantages of parenteral preparations. Injections cause pain at the site of injection. The trained person is required to administer the drugs. The administration of a drug through wrong route of injection may prove to be fatal or dangerous. It is difficult to save a patient when overdose is given. There are the chances of sensitivity reactions or allergic reactions of a drug by an individual. These reactions are sometimes very fatal and lead to the death. These are the disadvantages of a parenteral preparations. Now we will see the route of administration of parenteral products. The various routes of administration of parenteral products are used for giving the injections. Generally, there are the 10 routes by which the injection or parenteral products may be given. Now, the first one is the intradermal injection. Intradermal injection, it is also known as the intracutaneous injections. These injections are given in between the dermis and epidermis. Skin of the left forearm is usually selected for giving the injection. Generally, zero point 0.1 to 0.2 ml of parenteral solutions is injected by this route. The route is used for diagnostic purpose and for testing the sensitivity of injectables. Second one is the hypodermis injections. These injections are also known as the subcutaneous injections. These are made under the skin into the subcutaneous tissues. The volume of 1.0 ml or less is usually injected into the upper arm. This is the most popular route because it is convenient for patient and the doctor. Third one is the intramuscular injections. These injections are given into the muscular tissues. The muscles of the shoulder, thigh, or buttock are usually selected. Generally, volume up to 2 ml is administered by this route and should not exceed 4 ml at one site. Aqueous or oily suspensions and oily solutions can be administered by this route. Fourth one is the intravenous injections, which injections are made into the vein and therefore introduced directly into the bloodstream. The median basilic vein near the anterior surface of the elbow is usually selected because it is easily located and connects with the major veins of the arm. Large volume of parenteral solutions ranging from 1 ml to 500 ml or more than that can be injected. The parenteral solutions should be isotonic with blood if the volumes of more than 15 ml should be injected. The suspensions and oily injections cannot be injected by this route. Then the fifth one is the intra-arterial injections. These injections are similar to the intravenous injections and are occasionally used for an immediate effect in a peripheral area. These injections are directly given into the artery. Sixth one is the intracardiac injections. These injections are given into the heart muscles or ventricle in an emergency only. For example, as a stimulant following cardiac arrest. Seventh one is the intrathecal injection. These injections are made into subarachnoid space that surrounds the spinal cord. This route is used for spinal anesthesia. Another one is the eighth one, intracisternal injections. These are given in between the first and the second cervical vertebrae. These injections are used to withdraw CSF means cerebrospinal fluid for diagnostic purpose. Ninth one is the peridural injections. These injections are given between the dura mater and the inner aspects of the vertebra. So, it is that portion of vertebral canal not occupied by the dura mater and its contents. This route is sometimes used for giving spinal anesthesia in special cases. Tenth one is the intra-articular injections. These injections are given into the liquid that lubricate the articulating ends of bones in a joint. 11th one is the intracerebral injections. These injections are given into the cerebrum. Here in this diagram you can see the various angles which are used for the insertion of parenteral injections, in which you can see the intramuscular injections, subcutaneous injections, intravenous injections, intradermal injections, and also the degree how they are inserted or angles that are given in this image. Now, a general requirements for parenteral dosage forms. The formulation of parenteral products involves a careful considerations such as they requires stability, sterility, free from pyrogens, free from foreign particles, isotonicity, specific gravity, and chemical purity. Now, first of all, we will see the stability. The stability of a parenteral preparation is very important. The physical as well as chemical stability of parenteral preparation must be maintained during storage. Second one is the sterility. The parenteral preparation should be free from all types of microorganisms and aseptic conditions are required to be maintained during the preparation of parenteral product and its administration. The parenteral product must pass the test of sterility. Third one is the free from pyrogen. The parenteral preparations should be free from toxin and pyrogens. It is necessary that parenteral products must pass the test for pyrogen because contaminated parenteral product causes rise in a body temperature after its administration. Fourth one is the free from foreign particles. The parental products should be free from foreign particles such as dust and fiber. To ensure this, parental product must pass the clarity test. Fifth one is the isotonicity. The parental preparations should be isotonic with blood plasma and blood fluids. It is very important in order to avoid any complications on the administration of parenteral product. Sixth one is the specific gravity. The parenteral products meant for intraspinal injections should have the same specific gravity as that of spinal fluid into which the same are to be injected. Seventh one, chemical purity. The parenteral products should be free from chemical impurities or it should be within certain limits as specified the monograph of that preparation in the pharmacopoeia. Now, the types of parenteral preparations. The parenteral preparations are classified as solutions or emulsions of medicaments suitable for injections, sterile solids, sterile suspensions, and transfusion fluids. The first one is the solution or emulsions of medicaments suitable for injections. These are commonly called as injection. The parenteral preparation in this form may be supplied in a single dose container or multiple dose container. Its volume varies from 0.5 ml to a liter. Example, atropine sulfate injections, dextrose injection. Second one is the sterile solid. Drugs which are not stable in solution are prepared and supplied as a dry sterile solids which are dissolved in a suitable solvent immediately before its administration. Example, benzyl penicillin G sodium injection. Third one is the sterile suspension. These are the sterile suspensions of drugs in a suitable solvent which are administered by intramuscular route. Fourth one is the transfusion fluid. These are the parenteral solutions which are administered by intravenous route. They are generally used for nutrition and to maintain the electrolyte balance. Example, ringer solution, dextrose injection, sodium chloride injection, etc. Now, starting with the formulation of parenteral preparations. The formulation of parenteral preparations need careful planning. thorough knowledge of the medicaments and adjuvants to be used. The excess use of adjuvants in parenteral products should be avoided as some of these may interfere with the drug. In the preparation of parenteral product, the various substances are added to make a stable preparation, such as first one is the vehicle and second one is the adjuvants. Now we will see the first one, vehicle. Vehicles, there are two types of vehicles which are commonly used in the preparation of injection: aqueous vehicle and non aqueous vehicles. First one is the aqueous vehicles, in which water is used as a vehicle for majority of injections because water is tolerated by a body and is safest to administer. Second one is the non-aqueous vehicles. Non-aqueous vehicles are commonly used are oils and alcohols. Fixed oil such as arachis oil, cottonseed oil, almond oil are used as a vehicles. Ethyl alcohol is used in the preparation of hydrocortisone injections. Propylene glycol is used as a vehicle in the preparation of digoxin injections. These are the solvent as well as to increase the stability of certain preparations. Now, the second one is the adjuvants. In adjuvants, various substances are added to increase the stability or quality of the product. The adjuvants should be used only when it is absolutely necessary to use them. While selecting the additives, care must be taken that they should be compatible both physically and chemically with the entire formulation. They should be added in a minimum possible quantity. The various adjuvants are commonly used in the preparation of stable parental preparations are solubilizing agents, stabilizers, buffering agents, antibacterial agents, chelating agents, suspending, emulsifying and wetting agents, and tonicity agents, or it is also known as the tonicity factors. Now, the another point is the units of concentration. The concentration of ingredients in parenteral products may be expressed in a number of different ways. First one is the weight per unit volume. Example, atropine sulfate injection, which is given as 600 micrograms per ml, or prednisolone injections, which is given 25 micrograms per ml. Second one is the percentage weight by volume or weight per volume, in which a dextrose intravenous infusions are given 5% weight by volume. Sodium chloride intravenous infusions are given 0.9% weight by volume. Third one is the millimoles per unit volume. The examples are sodium bicarbonate intravenous infusion is given 150 millimoles each of sodium and hypocarbite per liter, and potassium chloride solution strong 2 moles. Now, the containers and closures which are used for parenteral preparations. The containers used for parenteral preparations or products are either made up of glass or plastic containers. The pharmacopoeia requires the various conditions for a container and closure to be used for parenteral preparations. These conditions are as follows. First one, it should not yield foreign substances to the product. Second one, it should be sufficient transparent to allow visual inspection of content in it. It should not have any adverse effect on the product. Fourth one, it should prevent diffusion in or across the walls. The following types of the glasses are used in the preparation of parenteral products or as a containers and closures of a parenteral products. First one is the borosilicate glass or natural glass. Second one is the soda lime silicate glass with hydrolytic resistance. And third one is the soda lime glass with only moderate hydrolytic resistance. These glasses are also known as the type 1 glass, type 2 glass and type 3 glass. Type 1 glass means what? It is called as the borosilicate or neutral glass, which offers high hydrolytic resistance due to its chemical composition. It can withstand autoclaving and weathering. Type 2 glass means a soda lime silicate glass with high hydrolytic resistance, in which it is treated with a moist sulfur dioxide at high temperature. The containers made from this glass are suitable for most acidic and neutral aqueous preparations. Third one is the type 3 glass, which is a soda lime silicate glass with only moderate hydrolytic resistance. These containers are made from this glass are generally suitable for non-aqueous preparations and powders for reconstitution immediately prior to the administration. Now, the processing of parenteral preparations. The following steps are generally involved in the processing of parenteral preparations. There are the 9 steps. First one is the cleaning of container closures and equipments. All the container closures and equipments which are required during preparation of parenteral product should be thoroughly cleaned with detergents, and washing is done with a tap water. Then it is followed by clean distilled water and finally rinse with the water for injection. Rubber closures are washed with a hot solution of 0.5% sodium pyrophosphate in a water. The closures are then removed from a solution, washed with water, followed by rinsing with filtered water for injection. On a small scale, washing is done manually, but on a large scale, automatic washing machines are used. Second one is the collection of material. The various ingredients of the formulation of parenteral preparations are weighed and collected in the preparation room. The raw material required in the preparation of the parenteral product should be pure. Water for injections free from pyrogen and microorganisms are used in the preparation of parenteral product. Third one is the preparations of parenteral product. The pharmacist should decide the order of mixing and exact method of preparations to be followed before preparing the parenteral product. The parenteral preparations must be prepared under strict aseptic conditions. Fourth one is the filtration. The parenteral solutions so formed is passed through the bacteria proof filter such as filter candle, seeds filter, membrane filter and sinter glass filter. Then filling of preparation of final container. The filter product is filled into final containers such as ampoules, vials or transfusion bottles which are previously cleaned and dried. Ampoules are used for filling single dose whereas vials are used for multi doses. Bottles are meant for filling transfusion fluids. On small scale, filling is done manually or by hypodermic syringe or needle. On large scale, the filling is done by automatic filling machine. Then the sealing of container. Sealing should be done immediately after filling. Ampoules are sealed manually on a small scale by rotating the neck of the ampoules in the flame of Bunsen burner. But on a large scale, ampoule sealing machine is used in which tip of ampoule is fixed to seal it. The vials and transfusion bottles are sealed by closing the opening with rubber closures. The rubber closures are held in a place by crimping the aluminum caps, which is done manually or by mechanically means. Then the sterilization. The preparations should be immediately sterilized after sealing in its final containers. The sterilization is done by one of the methods of sterilizations which depends upon the nature of medicaments present in a parenteral preparations. Then evaluation of parenteral preparations. The finished parental products are evaluated by a various tests such as sterility test, clarity test, leakage test, pyrogen test and assay. Then labeling and packaging. Labeling and packaging after evaluation of the parental preparations, these ampoules and vials are properly labeled and packed. This label should contain the name of preparation, quantity of preparation, manufacturing license number, batch number, date of manufacture, date of expiry, storage condition, retail price, and manufacturer's address. Now we will see the evaluation of parenteral preparation. How the parenteral preparations are evaluated. Now, the first test is the sterility test, or the evaluation is done by sterility test, clarity test, leakage test, pyrogen test, and assay. Now, we will see the first one, a sterility test. After sterilization, the test for sterility is most reliable method for determining whether the particular lot of a material is sterile. Test for sterility is intended for detecting the presence of viable forms of bacteria, fungi, yeast in a substances. The test it is carried out by either membrane filtration method or by direct inoculation method. Now, the principle of sterility testing. The test is based on a principle that if bacteria or fungi are placed in a medium which provides nutritive material and water and kept at a favorable temperature, the organism will grow and their presence can be indicated by turbidity in the clear medium. Now, the various steps involved in a sterility testing, that are the selection of sample size, selection of quantity of product to be used, method of testing, observation and results. Now, the second method is the clarity test. Clarity test is performed to ensure the parental products which are free from foreign particles. Each parental preparation in its final container is subjected individually to a visual inspection to exclude the possibility of foreign particles. The unlabeled conditions are held by the neck against strongly illuminated black and white screen. White screen is used for dark colored particles and black screen is used for the detection of light colored particles. Leakage test. This test is performed only for ampoules which have been sealed by fusion to ensure that there should not be any leakage in them. Leakage test is performed in a vacuum chamber. The ampoules are dipped in a 1% of solution of methylene blue in vacuum chamber and vacuum is applied. When vacuum is released, the color solution will enter the ampoule with defective sealing. The presence of dye in ampoule confirms that leakage and hence rejected. Vials and bottles are not subjected to this test because the flexibility of rubber. Another one is the pyrogen test. This test is done as per the IP 2007 to check the presence or absence of pyrogens in all aqueous parenteral preparation. The test involves the measurement of the rise in a body temperature of rabbits following intravenous injections of a sterile solutions of a parenteral preparations being examined. Rabbits are used to perform this test because their body temperature increases when pyrogens are introduced into their bodies by parenteral route. Assay. Assay is performed according to the method given in the monograph of that parenteral preparations in the pharmacopoeia. Assay is done to check the quantity of medicament present in the parenteral preparations. Now, starting with the production facilities. The manufacture of parenteral preparations required special precautions and facilitates in order to maintain sterility and freedom from particulate matter. The production area where sterile preparations are manufactured, it is divided as in a 5 sections such as cleanup area, preparation area, aseptic area, quarantine area, and finished and packaging area. Cleanup area, it is not an aseptic area. All the parental products must be free from foreign particles and microorganisms. The area and atmosphere of the room where manufacturing of parental products are done must be free from dust, fiber, and microorganisms. Preparation area, in this area, the ingredients of the parental products are mixed and preparation is made for filling operation. Although it is not essential that this area should be aseptic, but strict precautions are required to prevent any contamination from the outside. Third one is the aseptic area. From preparation area, the parenteral formulation will be transferred to the aseptic filling area. The parenteral preparations are filtered, filled into the final containers and subsequently sealed. The entry of personnel into aseptic area should be through an airlock. To maintain sterility, specially trained persons should be selected to work. They must be orderly, neat, and reliable. They are required to wear sterile clothes and should be subjected to physical examination at regular intervals to ensure that they are not carrier of any infectious disease. There should be minimum movement in the aseptic area. Fourth one is the quarantine area. A batch of parental products, after its filling, sealing, and sterilization, are held up in this area. The random samples of parental products of different batches are in the analytical laboratory for its evaluation. Those parental products which passes all the quality control tests are transferred into finishing or packaging area. Finishing and packaging area. The parental products are properly labeled and packed. Proper packaging is essential to provide protection against physical damage from transportation, handling, and storage. The label container should be packed in a cardboard or plastic containers. Ampoule should be packed in a partitioned box. The packed parental products are temporarily stored in its finally disposed of. Now we will see the particulate matter monitoring. All the parental products must be sterile, free from pyrogen and particulate matter. Now, particulate matter means what? There are the various sources of particulate matter which may interfere into the various parental products. So, to check the particles which are present, there are the various methods which are used for checking, such as visual method, Coulter counter method, filtration method, and light blockage method. Now, we will see the visual method. Visual method, it is an old and reliable method. According to this method, the filled containers are examined against strong illuminated screen by holding the neck and rotating it slowly or inverted it to exclude the possibility of foreign particles. Coulter counter method. The method is based on the principle that increases in resistance is observed between two electrodes. As the particle approaches and passes through the orifice, an electrolyte is required to be included in the preparation before its evaluation. The particle with diameter below 0.1 micrometer can be detected by this method. Filtration method. It is the method which is meant for counting particles in hydraulic fluids. The liquid sample is passed through the filter and the material collected on the surface of the filter is examined under microscope. Light blockage method. The method gives an automatic evaluation of particulate in hydraulic oil. It allows a stream of the fluid under test to pass between the bright white light source and photodiode sensor. It is possible to detect the cross sectional area of the particle in this instrument because it blocks the path of light, and size of a particle is considered as a diameter for the circle of its equivalent area. Here we have seen the chapter sterile products. Thank you very much.